Hi students, in this video we're going to be talking about cellular respiration. It's a pretty intimidating topic. It's very complicated. There's a lot going on. But one thing I love about cellular respiration is that it's so complicated yet so elegant in that everything in the cell just works together to keep the organism alive. And you know, the whole purpose of cellular respiration is to make ATP. We're going to be talking about the mitochondria, which everybody knows as the powerhouse of the cell. But what exactly about the mitochondria enables it to make ATP in such an amazing, sufficient, efficient manner? Okay, so that's what we're going to be talking about today. I've got my coffee here to keep me energized. Hope you guys are doing well today. I'm just going to go ahead and minimize myself. All right, so let's talk about cellular respiration. In our textbook, this is going to be chapter 9. I think in this video, I am going to be like skipping some slides because I don't want to go too long in the video, and I kind of want to just get to the meat and potatoes of what we're talking about in respiration. So in cellular respiration, we're basically taking chemical energy in food and we're converting that energy or using that energy when we break it down to make ATP. Okay, so yeah, living things need to make ATP because life is work, right? We need energy to make biomolecules, to grow, develop. We need ATP, we need energy to reproduce, for movement, active transport, right? Like the sodium potassium pump, and also temperature regulation. You know, we are endotherms. We maintain body temperature internally, and so we need ATP to do that. Okay, so what's the whole point of cellular respiration? It's to make ATP. So keep that in mind. That's like the big picture of cellular respiration. So an overview of cellular respiration, it happens in 3 stages. The first stage is called glycolysis, and glycolysis part does not require any oxygen gas, so it's considered to be anaerobic. Okay, anaerobic means without oxygen. And this is where a little bit of ATP is made, and it's made in the cytosol. So glycolysis takes place in the cytosol. The last 2 stages of cellular respiration is we've got the pyruvate oxidation and Krebs cycle. They're actually grouped together. This takes place in the presence of oxygen, and it takes place inside of the mitochondria. And then the 3rd stage of cellular respiration is called the electron transport chain. Okay, so yes, this is the part that requires O2, and it takes place in mitochondria. Okay, and this is just kind of an overall about where it takes place and what's being made. Okay, so we've got glycolysis, pyruvate oxidation and Krebs cycle, and the electron transport chain. I just want you guys to focus a little bit on what's made in each step. In glycolysis, let me bring up my pen. Okay, in glycolysis, we're making just a little bit of ATP, 2 ATP. In the Krebs cycle, we're making a little bit of ATP. The majority of ATP is made here in the electron transport chain. It says behind me, 26 to 28 ATP is made in the electron transport chain. Okay, but this is an overview of cellular respiration. 3 stages: glycolysis, pyruvate oxidation and Krebs cycle, and then the electron transport chain. But again, the whole point of cellular respiration is to make ATP. So let's discuss a little bit about mitochondria, right? Mitochondria, we have the outer membrane, and then you have the inner membrane. I'm just going to move me up here, okay? We've got the outer membrane, the inner membrane. The inner membrane is highly folded, right? And the folds are called cristae. The inside, the internal part of the inner membrane is called the matrix. And mitochondria, the outer membrane is smooth, inner membrane is highly folded. The space between the outer and the inner membrane is called the inner membrane space. Inside the center of the mitochondria is called the matrix. And remember that mitochondria, they have their own DNA and their own ribosomes. So it's got mitochondrial DNA. Sometimes it's called mtDNA, okay? And just so you guys know too, mitochondria are inherited by, you get it from your mom, okay? So mammals, we get our mitochondria from our mom. And so mtDNA is passed down from mom to child within the mitochondria. And they've got their own little ribosomes, remember? And this is one evidence of endosymbiosis. That's a theory that states that mitochondria were at one point in time free-living prokaryotes that were engulfed by a larger cell. And instead of being digested, they continued living within the organism, okay? So that's the structure of the mitochondria, which we need to talk about because most of the ATP is going to be made there. So you can see here that in this microscopic photo, we've got two mitochondria that are actually dividing. They're dividing just like bacteria do in a process. It looks like it's dividing by binary fission. Okay, so this is one mitochondria that's dividing into two. Here's another mitochondria that looks like it's dividing into two. And so this is another evidence of what we call endosymbiosis. Remember endosymbiosis? So let me just spell it out here. Endo, which means inside. Symbiosis is where you have two different organisms, species that are living in close association with each other. And the theory of endosymbiosis states that mitochondria used to be little tiny prokaryotes that were engulfed by larger cells, and instead of being digested and eaten by the larger cell, it remained alive inside. And now they're living, mitochondria are living within our cells, and they are now an organelle, okay? But they used to be little prokaryotes because they have their own DNA, their own ribosomes, etc. And within the mitochondria, you have the inner membrane. And remember, whenever we say membrane, we're thinking of a phospholipid bilayer. And the inner membrane also has proteins embedded in the phospholipid bilayer. These proteins are, some of them are enzymes, like this is ATP synthase, permeases, okay, which are going to help transport electrons. This is what we call the electron transport chain, or ETC for short. Within the inner membrane of the mitochondria, you have the electron transport chain because these proteins are going to move electrons through it. And we're going to talk about why this happens and how this happens for the purpose of making ATP. Okay? So yeah, the inner membrane of the mitochondria has the electron transport chain. And what's the advantage of having an inner membrane, highly folded inner membrane? It's to increase surface area to increase ATP production. Okay, you've got more surface area for membrane-bound enzymes and permeases to make more ATP. So how is ATP used as an energy source? Well, it's by phosphorylation, okay? ATP, and this is a diagram showing you ATP. This is the 3 phosphate groups, but let me just show you the way we did it in class. We've got the sugar, we've got our adenine, nitrogenous base, and then we've got 3 phosphate groups, okay? It's that last phosphate group at the end. You know, we've got strong covalent bonds here, but this bond, when it's broken, this phosphate group comes off, and it's what we call inorganic phosphate now, because this phosphate group is no longer attached to organic sugar or organic adenine, and so it's inorganic. And this inorganic phosphate, when it attaches to another molecule, it energizes that molecule, okay? It energizes that molecule. So like in this example, you know, the phosphate group is attached to this transport protein. It's going to transport molecules, maybe in active transport, okay? Or this phosphate group is attached to this motor protein, and the motor protein is going to move the organelle through the cell, okay? Or in this case, this inorganic phosphate is going to be part of a reactant that's going to become a product, okay? So ATP is used when that inorganic phosphate comes off of ATP. So yeah, ATP becomes ADP, and we've got the inorganic phosphate. It releases energy, okay? And when it releases energy, that inorganic phosphate is going to be used for other reactions. And when that inorganic phosphate attaches to another molecule, we call that phosphorylation. So like when something has an inorganic phosphate attached to it, it's been phosphorylated. It's got a phosphate group attached to it, it's energized, okay? Okay, and the phosphorylation process, that phosphate group, that inorganic phosphate, it doesn't just attach to the molecule. It needs to attach to a molecule using an enzyme. There's an enzyme called kinase that brings that phosphate group and brings the molecule that's being phosphorylated together. Okay, so remember what enzymes do? Enzymes can break bonds or make bonds. Well, this is an example of a kinase is an example of an enzyme that brings an inorganic phosphate group with the molecule that's being phosphorylated together, and it's making a bond. Okay, it's making a bond and phosphorylating something and energizing it. Okay, so here's ATP. These are the 3 phosphate groups right here. One of the phosphate groups is going to come off, and there it is. It's attached to this protein that is now phosphorylated. And how did this phosphate group get phosphorylated? There's a kinase, a protein kinase that brings the protein and that inorganic phosphate together, making that bond. And you can see that bond right over here. Okay, this is the bond that forms between the inorganic phosphate and that protein. So I like this diagram because it shows you like how an enzyme, in this case, it's breaking a bond and it's making a bond at the same time. Okay, so this is an enzyme. It's also called a kinase because in this case, it's going to phosphorylate ADP. Okay, you know, remember I said at the beginning that one of the purposes of cellular respiration is to make ATP. Well, in glycolysis and in the Krebs cycle, ATP is made this way where there's an enzyme and the part of the reactant is ADP. It gets phosphorylated and that's how you get ATP. Okay, so in glycolysis, this is what we call substrate level phosphorylation because ADP and this phosphate group, they're part of the substrate. They're part of the reactant, and that inorganic that phosphate group is going to join up with ADP to become ATP. And that's one of the products. Okay, when ATP is made using an enzyme, this is called substrate level phosphorylation. So remember, the whole point of cellular respiration is to regenerate or make ATP. Because you know, when your cells use ATP, right, you end up with ADP and inorganic phosphate. Well, the whole point of cellular respiration is to make ATP so the cell can continue using ATP as a source of energy. And there's 2 ways that ATP is made in cellular respiration. It's substrate level phosphorylation. That happens in the Krebs cycle and in glycolysis. But the majority of ATP is going to be made here in the electron transport chain. Okay, it's going to be, sorry about that line. The majority of ATP is going to be made here in the electron transport chain by oxidative phosphorylation. Okay? The reason why it's called oxidative phosphorylation, we'll be talking about that in a few minutes, but when something is oxidized, it loses electrons, and those electrons are going to move through an electron transport chain, and the final electron acceptor is going to be oxygen, and in that whole process, a bunch of ATP is going to be made. Okay? So when we say oxidative phosphorylation, that's happening on the electron transport chain. And the electron transport chain is going to be building up what we call a concentration gradient of protons. And at the end of the transport chain, there is an enzyme called ATP synthase. It's a really cool enzyme, ATP synthase. And as the protons or H pluses move down their concentration gradient, because there's like a whole lot of them here on this side, if there's like a whole lot of them on this side, they're going to move down their concentration gradient through ATP synthase. It's interesting because it's an enzyme that has like this the C10 subunit of the protein that rotates. It's like a rotor. As the H plus moves through it, it causes this subunit to rotate. And as it rotates, it causes the other subunits to rotate as well, that bring ADP and inorganic phosphate together and generates ATP. It's an amazing, amazing enzyme. Okay? We'll talk more about ATP synthase later. But how do we make ATP from fuels like food? Okay, how do we use it? Well, what we do is we digest large molecules into smaller ones. We break bonds. And when we break bonds, there's going to be a release of energy. There's going to be movement of electrons from one molecule to another. So as the electrons move from one molecule to another, energy is going to be released, okay? But also the electrons are going to be used to create a concentration gradient that's going to be used to make ATP. That's the electron transport chain. More on that later, okay? So as electrons move, they carry energy with them. That energy is stored in another bond. It's or released as heat or harvested to make ATP, okay? So what you can see here is this is what we call a redox reaction, okay? A redox reaction. Redox reaction, redox, okay, that stands for reduction, and I believe on the next slide we're going to be talking about this again. Reduction, oxidation, reaction, okay? So when something loses electrons, it's oxidized. Okay, it's oxidized. So down here you can see there's an arrow that's showing an electron. That's an electron. It moves over to the next molecule. This one gets reduced. I know that sounds very counterintuitive. Like if it gains an electron, it becomes reduced. It seems like when something gains something, it shouldn't be called reduced, right? Because that word reduce makes it sound like it's losing something. But in this case, it's gaining negative charges, okay? So maybe you can think of it that way. If it gains negative charges, it becomes more negative. So maybe it becomes more reduced in its charge, okay? So anyways, this molecule, it loses electrons, it becomes oxidized. The one that gains, it becomes reduced, okay? So this is a redox reaction because a molecule is giving up electrons to the other, it's oxidized, and then the other molecule, the receiving molecule, gets reduced. And that's what's happening in cellular respiration, okay? In cellular respiration, it's a catabolic reaction. We are moving electrons from the food that we eat, and as the electrons are moving, there's a release of energy that can be used to synthesize ATP. So at the bottom, it got kind of cut off down here, but glucose is the molecule that we're really going to be focusing on when we discuss glycolysis and the Krebs cycle, okay? Glucose sugar, it becomes oxidized, it loses electrons, and the molecule that's gaining those electrons is oxygen, okay? So glucose gets oxidized, okay? It gets oxidized, and then the O2 gets reduced. That's the molecule that gets reduced. It's going to gain electrons. And you can see that here in the form of hydrogen. These hydrogens, the electrons are going to move over to the oxygen in the form of hydrogens, okay? And you can see what happens here. If you remove the hydrogens, what two elements are you left with? You're left with carbon and oxygen, right? And then if you move the hydrogens over here to oxygen, what are you left with? Hydrogens and oxygens, water, okay? So this is the equation for cellular respiration. You've got C6H12O6, which is glucose, plus 6 oxygen molecules. It produces 6 molecules of carbon dioxide and then 6 molecules of H2O plus energy. And that energy is either lost as heat or that energy is going to be used to make ATP, okay? So yeah, oxidation, the C6H12O6 glucose is oxidized, the O2 is reduced, when the sugar gets oxidized, it becomes CO2, you guys. And this CO2 is the carbon dioxide that you're breathing out right now. You're breathing the carbon dioxide. Where did that CO2 come from? It came from the sugar that you just ate. Not just ate, but the sugar that you ate, okay? And then where does that water come from? Some water is being made in cellular respiration. It comes from the O2 that you're breathing in. So you breathe in oxygen, right? And some of the water that's made in cellular respiration comes from that oxygen, okay? But the whole point of cellular respiration is to make that ATP. And we get that from breaking down or oxidizing glucose. Waste product is CO2, and you make water. But the whole point is to make ATP. So earlier I had said that electrons are being moved, right? Electrons are being moved from glucose to oxygen. How does that happen? Is it just a one-step process? No, it's not a one-step process, okay? It's actually a multi-step process. So let me just go back to this diagram that shows you the equation for cellular respiration. So looking at an equation for cellular respiration, that glucose, we said that that glucose is going to become oxidized. It's going to lose electrons. How are those electrons going to be lost? Is it just a one-step process where it just transfers the electrons over to oxygen? No, it's a much more difficult process, okay? So those electrons from glucose, they're actually going to be moved to oxygen in very kind of complicated steps, okay? Multi-steps, many steps. So they're going to be transferred, though, using these two molecules called NADH. So actually NAD and FAD. So we'll just say NAD and FAD, okay? NAD and FAD are electron carriers. They're going to get electrons from glucose, and they're going to move them over to that oxygen in the electron transport chain. Okay, so one thing I should specify is that, glucose, when you eat glucose and it gets transported into your cells, it's in the cytosol, okay? It's in the cytosol where it's going to be first broken down in glycolysis, and that O2 is going to actually go into the mitochondria. That O2 is going to be in the mitochondria, okay? And NAD and FAD are going to take those electrons to the electron transport chain in the mitochondria. So they're taking the electrons and they're going to move them over to oxygen in the mitochondria. So the NAD and FAD are what we call electron carriers. I like to, use the analogy like they're the Ubers, they're the Lyft drivers of electrons, okay? They're going to take the electrons from glucose and then they're just going to transfer it over to the electron transport chain. They're the Uber drivers, they're the Lyft drivers of electrons. You don't need to know the structure of NAD and FAD, but I just want to bring up the fact that they are a type of nucleotide. Just like how ATP is a nucleotide that's not used really for hereditary purposes or DNA purposes. ATP is a source of energy, but NAD and FAD are used for something else. They're electron acceptors or electron carriers. They carry electrons. So the electron transport chain, this is again what it looks like. It's embedded. The electron transport chain is the inner membrane of the mitochondria. You've got embedded proteins and enzymes in there. Okay, so the flow of electrons from glucose get moved to NAD and FAD. NAD and FAD are going to carry those electrons to the electron transport chain, and then oxygen is going to be the final electron acceptor. And why is electron the final electron acceptor? Remember we said that oxygen is electronegative. Oxygen is the final electron acceptor because oxygen is electronegative. It loves electrons, right? It's going to attract the electrons. So electron transport chain embedded in that inner membrane of the mitochondria where you've got a series of proteins where the electrons are going to flow through it. You can see in this diagram, the yellow arrow shows you the pathway of the electrons. And the final electron acceptor is O2, and it's going to combine with those electrons to make water. All right. So let's talk about glycolysis, and the name kind of says it all. Glycolysis, glyco is referring to glucose, lysis means break, okay? And this is all taking place in the cytosol. You guys, basically what's happening here is I'm just gonna simplify what glycolysis is in a diagram, and it's a lot more complicated than this, okay? Glucose is a 6-carbon sugar, correct? It's got C6H12O6. And for the purposes of simplicity, I'm just going to draw the 6 carbons this way, 4, 5, 6. So this is my 6 carbons in glucose. In glycolysis, all we're doing is taking glucose and breaking it down the middle. Okay, breaking it down the middle, and you end up with 2 molecules. You end up with 2 molecules that have 3 carbons each, and each one is called pyruvate. Okay, pyruvate. And this is happening. The breaking of glucose happens in 9 steps. It's not just one simple step or just cutting glucose in half. It's actually 9 steps where you're going to end up with pyruvate, okay? You're going to end up with 2 pyruvate. And pyruvate are 2, you've got the 2 pyruvate are 3 carbon sugars. In the 9 steps, okay, you do make 2 ATP. Remember, the whole point of cellular respiration is to make ATP. So you do make a little bit of ATP in glycolysis. You make NADH, okay? Remember, NAD is going to take some electrons. And when it picks up electrons, we call that NADH. Where is it going to take the electrons? To the electron transport chain, okay? And 2 pyruvate. The 2 pyruvate is a 3-carbon compound, which is actually going to go into the mitochondria if oxygen is there, okay? Actually, there are 4 ATP that's made in glycolysis, but there's a step in glycolysis that actually requires ATP, and so you use up 2 ATP in glycolysis. So there's a net ATP of 2. Okay, there's a net of 2. And so how is ATP going to be made in glycolysis? It's called substrate level phosphorylation, because you have an enzyme that's bringing that phosphate group to ADP and making ATP. Okay. Glycolysis in Krebs cycle generates or makes ATP this way using an enzyme. So these are the 9 steps that I was talking about, okay? You start off with glucose, you end up with 2 pyruvate, 1 here, 1 here, okay? 9-step process. You'll notice that there's an enzyme for every step. Hexokinase is the first enzyme for the first step. Phosphoglucose isomerase, phosphofructokinase, aldolase, G3P dehydrogenase, phosphoglycerate kinase, phosphoglyceromutase, enolase, and then pyruvate kinase, okay? So there's 9 steps to go from glucose splitting into 2 molecules of pyruvate, glycolysis, okay, splitting of glucose. And for each step, right, there's an enzyme. So that's why I like to say in class that there's an enzyme for that. There's an enzyme for every single little step. It's just amazing. It's amazing how your cells make all of these different enzymes that have a specific purpose, okay? Like phosphofructokinase, it only does one thing, which is to convert fructose 6-phosphate into fructose 1,6-biphosphate, okay? Amazing. So hexokinase, it's a kinase. And what do we say what a kinase is? Remember, a kinase is an enzyme that phosphorylates, okay? And in this case, it's going to take that, third phosphate group from ATP, and it's going to slap it on to the glucose. There it is. There's our phosphate group attached to our glucose. So that glucose is now phosphorylated, and we call that glucose 6-phosphate. The reason why we call it glucose 6-phosphate is because that phosphate group is attached to the carbon number 6, okay? Carbon number 6. This is carbon number 1. Okay, this is carbon number 1. This is carbon number 2. This is carbon number 3. This is carbon number 4. This is carbon number 5. And then this is carbon number 6. Carbon number 6 has a phosphate group attached to it. That's why the name is glucose 6-phosphate. phosphate group is attached to the 6th carbon. And then now we have another enzyme called phosphoglucoisomerase, okay? Notice how it says isomerase? Remember what isomers are? Isomers are basically molecules with the same molecular formula but different structure. So what it is it's just taking glucose 6-phosphate and it's rearranging the atoms just slightly so that it becomes a fructose 6-phosphate. You know, glucose is shaped like a hexagon, whereas fructose is shaped like a pentagon. So it changed the bonds around a little bit, so you end up with a pentagon. And then ATP is going to be used again here, okay? ATP is going to be used here again because that 3rd phosphate group is going to come off. Okay, this is ADP, by the way, ADP. The ATP becomes ADP because there's another kinase involved here. That phosphofructokinase is going to phosphorylate that fructose 6-phosphate. And so now we have fructose 1,6-biphosphate because you've got a phosphate group attached to the carbon number 1 and you've got a phosphate group attached to carbon number 6 right here and right here, okay? I'm not going to go over every single little step because I bet you're probably like, what is going on here? This is so overwhelming. Luckily for you guys, you don't need to remember all the names of the intermediate molecules. These are the intermediate steps, okay? You don't need to know the names of these intermediate molecules, nor do you need to know the names of all of the different enzymes involved, but you should definitely know or recognize what kinases do in general. They phosphorylate, okay? So again, there's 9 steps involved here until we get to pyruvate. And I apologize, it got cut off here, but let me just redraw it. Okay, so pyruvate looks like this. It's a 3-carbon sugar. Well, not a sugar. It's a 3-carbon molecule. You've got 1, 2, 3. Okay, this is what pyruvate looks like. And when glucose is split in half, you end up with 2 of these guys. Okay. Okay, so glycolysis in 9 steps, we're taking glucose and we're cutting it into pyruvate, okay? And in the whole process of glycolysis, one thing I should point out here is I should point out where the NADH and the ATP are made. So remember this intermediate, it's called 1,3-biphosphoglycerate, okay? Well, there's an enzyme called phosphoglycerokinase. Kinase is an enzyme that phosphorylates. In this case, it's going to phosphorylate ADP. And when it phosphorylates ADP, you end up with ATP, okay? And so you're going to make 2 ATP here. And then here's another 2 ATP that are made via pyruvate kinase. NADH is made here, okay? NAD, remember, is an electron carrier. It's going to be it's going to be reduced. Sorry, I almost said the wrong thing. It's going to be reduced, meaning it's going to gain electrons. And when it gains electrons, it becomes NADH. Okay? And remember what NAD and FAD do? They take electrons, and then they take the electrons, they're the Uber drivers. They're going to take the electrons to the electron transport chain. Okay? So, maybe I should draw it here. So like the NAD, okay, so this is the electron transport chain somewhere over here. It's embedded in the inner membrane of the mitochondria, ETC, it's all over, okay? So we've got NADH. It's carrying the electrons from glycolysis to the electron transport chain, okay? And in glycolysis, we've made a net of 2 ATP. We made 4, but then 2 were used, so it's 4 minus 2, and you end up with 2 extra ATP. Okay. Now we're going to talk about Krebs cycle, or it's called citric acid cycle as well, because citric acid is the first product of the Krebs cycle. Pyruvate, if oxygen is available in the cell, then it's going to enter the pyruvate is going to be shuttled into the matrix. Remember, this is the matrix of the mitochondria. It's the center of the mitochondria. So if oxygen is present, pyruvate will enter the mitochondria. Pyruvate is oxidized. It's going to lose electrons, okay? And it's going to be oxidized in 3 steps. So here's our pyruvate. the minute it enters the mitochondria, by the way, there's enzymes in all of these steps, but we're not even showing them, okay? There's an enzyme that's going to break off, okay? See this right here? Break off this portion of pyruvate, that's CO2, right? That's carbon and 2 oxygens. So CO2 is going to be removed, but then this leftover molecule is going to be oxidized. It's going to be oxidized. It's going to remove, there's going to be a removal of electrons, passing the electrons to NAD to make NADH. And you guys, where is that NADH going to go? Straight to the electron transport chain. It's going to go to the electron transport chain. And then the third step, there's going to be an enzyme that brings, okay, that molecule together with coenzyme A. Coenzyme A. This is what I'm talking about. Coenzyme A. So coenzyme A and whatever's left over from the pyruvate is going to make acetyl-CoA. Okay, so this is acetyl-CoA. So acetyl-CoA is a 2-carbon molecule. 1, 2. It's a 2-carbon molecule that's going to enter the Krebs cycle. So like this is the first part of the pyruvate oxidation and Krebs cycle. Okay, so we've got acetyl-CoA, and I know this looks very intimidating. Okay, acetyl-CoA is going to combine with oxaloacetate to make the first intermediate product, which is called citrate or citric acid. Okay, again, you don't need to memorize all of the intermediates, and it's not even shown here, but every single step requires an enzyme, and it's not even shown here. Okay, I just want to point out to you guys that acetyl-CoA is a 2-carbon molecule. Okay, we want to count the carbons. It's a 2-carbon molecule. Oxaloacetate is a 4-carbon molecule. You can see that 1, 2, 3, 4. Okay, and when they come together by an enzyme is going to bring them together, you end up with a 4-carbon molecule. 6-carbon molecule, because 2 plus 4, Mishim, 2 plus 4 is 6. 1, 2, 3, 4, 5, 6. Citrate is a 6-carbon molecule. Okay, and then citrate, there's an enzyme involved with this step. Okay, where there's a little bit of shuffling happening between the molecules, and it loses water. It becomes isocitrate, like an isomer. It's just changed its structure slightly. It's still a 6-carbon molecule. But then the next step, which requires a couple of extra enzymes here, okay? Isocitrate is going to lose a CO2. Okay, it's going to lose. See this right over here, that CO2? It's gonna be lost as waste, carbon dioxide. That's what you guys are breathing out right now. And then isocitrate not only loses that CO2, but it's going to be oxidized. It's going to give electrons to NAD. NAD is going to become NADH. It is an Uber driver. It's the Lyft driver. It's going to take the electrons where? Straight to the electron transport chain. Okay? And then now you end up with a 5-carbon molecule. It's called alpha-ketoglutarate. Alpha-ketoglutarate is also going to be oxidized, meaning it's going to lose electrons, and then also it's going to lose a CO2. See that CO2? That CO2 that you're breathing out right now? That CO2 that you're breathing out right now comes from these 2 steps in the Krebs cycle. And then NAD is going to take electrons to the electron transport chain. You end up with a 4-carbon compound called succinyl-CoA. Succinyl-CoA also gets oxidized, okay? And in this step, there's GDP. Okay, GDP, remember, like guanine diphosphate. It's a nucleotide in this case, okay? Where GDP gets phosphorylated, and then it passes the phosphate group to ADP to make ATP. Okay, and it got cut off at the bottom. But this is the place. This is the only place in the Krebs cycle where ATP is actually made. That coenzyme A also is removed. Okay, coenzyme A is removed. So succinate, okay, is still a 4-carbon compound. All right. Then FAD is going to come up, and it's going to take electrons from succinate. So succinate is going to be oxidized. FAD is going to be reduced. You guys, where does this FADH go? Straight to the electron transport chain. Okay. Then you get fumarate, which is still a 4-carbon sugar. You add, using an enzyme, adding water, you get malate, which still is a 4-carbon compound. Malate, okay, malate gets oxidized. It loses electrons, giving electrons to NAD. NADH, where is it going to go? Straight to the electron transport chain. Okay. Then malate becomes oxaloacetate, still a 4-carbon sugar, 4-carbon compound, I should say. So oxaloacetate is what's being recycled here. It combines with acetyl-CoA to make a 6-carbon compound called citrate. It just goes, you guys, Krebs cycle keeps on going over and over again. Anytime acetyl-CoA comes in, it becomes citrate, isocitrate, alpha ketoglutarate, succinyl-CoA, succinate, fumarate, malate, etc. And in the process, you're making 1, 2, 3 NADHs. You're making 1 FADH2. You're making 1 ATP. Okay? So I'm saying 1 ATP, 1 FADH2, 3 NADHs, but in actuality, guys, how many acetyl-CoAs are coming in at a time? It's actually 2, because remember 2 pyruvate were made in glycolysis. So that means that 2 pyruvate from glycolysis is responsible for 2 acetyl-CoAs. So really, 1 glucose molecule, from 1 glucose molecule, you get 2 pyruvate, 2 acetyl-CoAs. So then you have a total of 6 NADHs, not 3. You have a total of 2 FADH2s, and then you have 2 ATP that's made in the Krebs cycle that comes from that 1 glucose molecule. Okay, so really you get, it's oh, I forgot to mention the carbon dioxide. It's really 4, okay, really 4 carbon dioxide molecules. It's really 6 NADHs, 2 ATPs, and 2 FADH2s, because for each acetyl-CoA, it's really 2 acetyl-CoAs for each glucose molecule that we started with, okay? All right, so I like this diagram because it kind of gives you a big picture. We were just talking about the citric acid cycle, okay? You've got CO2s being, that's waste, but then you've got 2 ATP that's made here, right? And then you have the 6 NADHs and the 2 FADH2s that are made in the Krebs cycle. They go, where do they go? They go straight to the electron transport chain. Electron transport, okay? And then remember the NADH that was made in glycolysis? It goes straight to the electron transport. Okay, so NADH, FADH2s, those are the electron transporters or carriers. They carry electrons straight to the electron transport chain. In the meantime, you end up with 2 ATP from the Krebs cycle, 2 ATP from glycolysis. All right, so far, the cell has made 4 ATP total. But now we're going to talk about the electron transport chain. Oxidative phosphorylation is where a majority of ATP is made. Okay, so actually, I forgot to mention where this ATP is made. There's an enzyme, right? There's an enzyme that brings a phosphate group, takes the phosphate group from GTP, adds it to ADP, and you get ATP. Okay, so it's still substrate level phosphorylation that's happening here in the Krebs cycle because it's an enzyme that brings that phosphate group to ADP to make ATP. So the electron transport chain, again, where is it? It's embedded in the inner membrane of the mitochondria. And what's the point of having a highly folded inner membrane or all that cristae? It's to have as many ETCs or electron transport chains embedded in there so you can make as much ATP as you can, okay? So again, this is what it looks like. So the NADH and the FADH2s that came from glycolysis and FADH2, which came from Krebs cycle, they're going to drop off the electrons, okay? So that's what the yellow arrow shows. It's the pathway of the electrons. So electrons are going to move through this electron transport chain, and then it's going to be received by oxygen. Oxygen plus 2 protons, which is H+, and electrons make water. So water is a waste product of cellular respiration. All those electrons that were carried from glycolysis and Krebs cycle are going to end up going to oxygen in the electron transport chain to make water. Okay? So what's happening here? What's happening when the electrons are going through the electron transport chain? We've got 1, 2, 3. We have these 3 proteins that are embedded in the inner membrane, okay? And these proteins are very special because when the electrons go through them, electrons give them energy, okay? That's what electricity is, right? Isn't electricity basically the movement of electrons? And so as the electrons move through this protein, it's electrified, it's energized, and what it's going to do is these are what we call proton pumps, okay? Remember when we went over active transport and we said, if it's a protein that's called a pump, that means it's actively transporting whatever it's transporting, okay? These 3 proteins are called pumps, okay? They're not, this one is a protein complex 3, and this is 4. That's because there's other ones in between, okay? But there's 3. In the electron transport chain, there's 3 proton pumps. What they're gonna do is when electrons go through them, they're gonna pump protons, or H+, from the matrix, okay? So this is the matrix, right? The center of the mitochondria. And they're gonna pump the proton into the intermembrane space, the inner membrane space, okay? So I'm just gonna maybe use a different color here. I'm gonna draw a mitochondria just so you guys can see the bigger picture. So I've got my folded inner membrane, okay? And I've got my ETC embedded in that inner membrane. So what's happening is that these pumps are pumping protons, or H+, into the inner membrane space. So you guys, there's gonna be a buildup, all right? There's gonna be a buildup of protons in the inner membrane space. There's going to be a concentration gradient. Remember that term? Concentration gradient. Meaning there's a lot of something. There's a lot of solute on one side of the membrane. Okay? So we've got a high concentration gradient of protons in the inner membrane space. So NADH drops off electrons at the first proton pump. That electron is going to move through it. It's going to power that proton pump to pump protons into the inner membrane space. And then it gets transferred to these other coenzymes, coenzyme Q, okay? And coenzyme Q is going to transport, it's a type of lipid, by the way. It's going to transport that electron to this second proton pump. As the electron goes through that second proton pump, what is it going to do? It's going to power it, give it energy to pump protons to the inner membrane space. And then the electron is going to move to cytochrome C, okay, which is it's a protein, but it's an accessory protein. It's not a pump. It's just going to hand off the electron to the third proton pump. As the electron goes through the third proton pump, what is it going to do? Energize it to pump protons into the inner membrane space. And then the final electron acceptor is going to be oxygen. Oxygen is going to receive it along with 2 protons. It's going to make water. Okay? So the electrons that came from NADH, which came from glycolysis and the Krebs cycle, are going to move through this chain of proteins or proton pumps until finally it gets to oxygen. As it goes through these proton pumps, it energizes them to pump protons into the inner membrane space, creating a concentration gradient. And this concentration gradient is going to be super important because it's setting it up. You guys, we're setting up the scenario. It's going to set up a concentration gradient so that it can be used to make ATP. Because remember, ATP, making ATP is the whole point of cellular respiration. Okay, I haven't talked about FADH yet. In this diagram yet. FADH2 is going to transport and drop off the electrons here, okay, at this protein called protein complex 2, okay? It bypasses the first protein, it bypasses the first proton pump. FADH2 does not drop off electrons at the first proton pump. It drops it off here. The electrons go through the second proton pump, and then it goes through the third proton pump. So NADH is responsible for building up a higher concentration gradient because it goes through, the electrons from it go through three proton pumps, but FADH2 only, the electrons from FADH2 only go through 2 proton pumps, okay? But they're both equally very important because they're going to help build up this concentration gradient, okay? So let's talk about why is this concentration gradient being produced. Well, if there's a high concentration gradient of H plus in the inner membrane space, where do they want to diffuse to? Where do they want to go? They want to go back into the matrix, okay? So these protons, they want to go back in. They want to diffuse back into the matrix. Well, by diffusion, right? In this case, it's facilitated diffusion, everybody. It's facilitated diffusion. These protons are going to move down their concentration gradient through an enzyme called ATP synthase. And I'll be showing you some diagrams of ATP synthase. It's really amazing, okay? It's made up of several subunits, and it's like a rotor. As the H plus moves through them, it brings ADP and inorganic phosphate together to make ATP. It's amazing. It brings that, like enzymes, right? Remember how we said enzymes they're making bonds or breaking bonds? In this case, it's making bonds. It's bringing the two substrates together, the two reactants together, in this case, ADP and inorganic phosphate. It's bringing it together to make ATP, okay? So this is the electron transport chain. This first half, or first, I guess 3/4 of the electron transport chain, you've got these proton pumps. It's building a concentration gradient so that it can be used by ATP synthase to make ATP. I don't want people to get confused with electrons and protons and stuff like that. You guys, the electrons, they're just being used to energize proton pumps, but the final electron acceptor is oxygen to make water. But the protons are just being recycled, okay? So as the protons, the protons are not becoming, it's not becoming part of ATP, or, they don't combine with the electrons. Actually, they do combine with electrons to make water, okay? But the protons are just being recycled. Most of them are just being recycled. The H pluses, the protons, come down their concentration gradient, and then they get pumped back out, okay? They just get pumped back and around and around and around. Some of them will combine with oxygen and electrons to become water, but most of them just get pumped around and around, pumped out, diffuse back in, pumped out, diffuse back in, pumped out, okay, for the purpose of making ATP. So like I said, this is so complicated but amazing. It's so efficient, so elegant in the way that our cells make ATP. So this whole ATP synthase, remember how I was saying it's like a rotor? So I really like this diagram because you can see here that the H plus, it enters this part of the protein, the enzyme. By the way, if you've got a ton of, subunits, I don't know, I forget how many subunits there are that make up ATP. I'll look it up and I'll put it up on the screen, okay? But ATP synthase is an enzyme. Functionally, it's made up of many different parts. What level of protein structure is ATP synthase in? I hope you guys are saying quaternary, okay? Quaternary structure, because you've got all of these subunits that are coming together to become one functional enzyme, okay? So this proton, as it diffuses in, it locks into this position. It moves. It's like a water wheel, a rotor. It causes the rotor to go in like this clockwise direction, and then it comes out the other side. It diffuses back into the matrix. So remember, this is the matrix of the mitochondria. And as it causes this part of this enzyme to rotate, it causes this bottom part to rotate. This bottom part of the enzyme brings ADP and inorganic phosphate together to make ATP. Okay? It brings them together, makes that covalent bond happen between ADP and inorganic phosphate. Voila, ATP is made. This is what we call chemiosmosis. It's a little, when we think osmosis, we think of the movement of water, but in this case, it's not really the movement of water, it's the movement of protons that's responsible for making ATP. So what is chemiosmosis? How should I describe what chemiosmosis is? It's the building up of a concentration gradient for the purpose of making ATP. Okay, so we've got the electron transport chain. There's 3 proton pumps. As electrons from NADH and FADH2, as the electrons move through the proton pumps, they end up pumping protons into the inner membrane space of the mitochondria, building up a concentration gradient. That concentration gradient can be used, right? Because those protons are going to diffuse, facilitated diffusion. They're going to move down their concentration gradient. As they move through ATP synthase is the enzyme that's going to bring ADP and inorganic phosphate together to make ATP. Okay, so that's chemiosmosis. It's building up a concentration gradient to make ATP. You do need to know how the concentration gradient is made, okay? You have to know that NADH, FADH2 are these electron transporters. They're going to drop off the electrons at the proton pumps, which are going to power those electrons, are going to power them, give them energy to do what they need to do, which is to pump protons into the inner membrane space, building up a concentration gradient that's going to be used. The protons are going to move down, facilitated diffusion, move down the concentration gradient through ATP synthase. ATP synthase is going to bring ADP and inorganic phosphate together to make ATP. So it's amazing what our cells do. Most of the ATP that's made in cellular respiration, mitochondria is a powerhouse of the cell, right? It happens like this, okay? It's amazing what our cells can do. And this is just showing you another diagram, a bigger diagram about what's happening here. And this is what we call oxidative phosphorylation. Okay, oxidative phosphorylation, because electrons are being moved, okay? Things are being oxidized, right? NADH and FADH2, they're being oxidized. They're losing electrons. They're passing electrons to these molecules, and the electrons are moving through them until finally they get to oxygen, which makes water in that process. And phosphorylation is happening as well here, right? Because that ATP synthase is bringing ADP and inorganic phosphate together to make ATP. So that's what we mean by oxidative phosphorylation, and majority of ATP is made in the electron transport chain. The ETC, electron transport chain, builds a proton gradient, which can be used to make, it's going to power that ATP synthase to make ATP, okay? So yeah, we did it. We set up a proton gradient, allow for the protons to flow through ATP synthase by facilitated diffusion, and that ATP synthase is going to make ATP. Okay, that facilitated diffusion as the protons move through ATP synthase, we call that the proton motive force, because it's like a rotor. As the H pluses move through it, it rotates these subunits of the ATP synthase. It powers it to bring ADP and inorganic phosphate together to make ATP, proton motive force. Let me see how long this video is. Let me see here. Oh, it's 4 minutes and 59 long. It's probably too long, but it's a really good video that shows you how ATP synthase works. You guys could look it up. I think it was made by Harvard University. ATP synthase in action. It's pretty cool, okay? The little yellow molecules are the protons. They're going through the ATP synthase, and ATP synthase is bringing ADP, sorry, and inorganic phosphate together to make ATP. Okay, so yeah, you guys can check that out on your own. So there's the, yeah, it's pretty cool. Okay, anyways. So this is a summary of cellular respiration, okay? Glucose breaks down into 2-pyruvate. The 2-pyruvate, if oxygen is available, it'll enter the matrix of the mitochondria where the Krebs cycle takes place. In the Krebs cycle, we're making 2 ATP, also 6 NADHs, 2 FADH2s that are going to enter the electron transport chain where majority of ATP is going to be made, okay? Also in glycolysis, you make 2 NADHs. They're going to go straight to the electron transport chain where it's going to be used for oxidative phosphorylation, making more ATP, okay? So I'm just going to circle the amount of ATP that's made in each part of cellular respiration. So summary, there's our chemical equation. You've got glucose plus oxygen. You get CO2 as a product. Remember, where is CO2 made? It's a waste product that happens or is produced in the Krebs cycle. Where is that water made? It's made in the electron transport chain. And about 38 ATP is going to be made total. Okay. Okay, so I'm just going to skip this because we do need to talk about fermentation now. You know, there are some organisms that only do glycolysis because they don't require a whole lot of energy. There are some organisms that can actually just live off of 2 ATP that's made in glycolysis. Glycolysis doesn't require oxygen. So, you know, there are some organisms that do anaerobic respiration. Anaerobic, again, means no oxygen needed. We call this fermentation. Fermentation catabolism. Remember catabolic reaction where we're breaking down? So we're breaking down glucose in the absence of oxygen. So let's talk about fermentation. And there's 2 types of fermentation. There's alcohol fermentation, where alcohol is made, specifically ethanol alcohol, like beer and wine. And there's lactic acid fermentation, where pyruvate is going to be converted into lactic acid, which is a 3-carbon compound, I should say, that's made in like mussels. Your mussels make lactic acid when you don't have enough oxygen during cellular respiration, okay? Pyruvate's a branching point, meaning pyruvate is made at the end of glycolysis. If there's no oxygen available, then fermentation is going to take place. If oxygen is available, then the pyruvate's going to enter the matrix of the mitochondria where it will go through Krebs cycle. Okay, so yeah, if there's no oxygen, then fermentation will take place, what we call anaerobic respiration. But if there is oxygen available, it's going to enter the mitochondria where it goes into the Krebs cycle. That's called aerobic respiration. So let's talk a little bit about some examples of fermentation or anaerobic respiration, okay? You know, bacteria and yeast, those are organisms that can take the pyruvate, which is a 3-carbon compound, and make ethanol alcohol and carbon dioxide as a waste product, okay? And so something that's really important to recognize here is that pyruvate turns into ethanol when it loses a carbon, okay, in the form of carbon dioxide, but also the pyruvate is going to become reduced. It's going to be reduced. Remember what we said about reduction? Reduction is when you gain electrons, okay? So what happens is that NADH is going to lose electrons, so the electrons are going to go from NADH to pyruvate, and NADH is now oxidized. It loses electrons, giving electrons to pyruvate, and that NAD plus can now be used for glycolysis. And remember, in glycolysis, they're making 2 ATP. So this step of fermentation is really important for bacteria and yeast because they need that NAD plus. They need to regenerate. They need to make NADHs. They need to make NAD pluses to go back to glycolysis so that at least 2 ATP can be made. Okay? Remember, fermentation, you only get 2 ATP in glycolysis, and it's enough for bacteria and yeast to survive. So that NAD plus needs to go back to glycolysis. In that process of glycolysis, they're making 2 ATP. Now and also bacteria and yeast, like humans use them for the purpose of making beer, wine, and bread. And you know, like when you slice into bread, you see all the little pockets, right? The empty pockets. Well, where did the pockets come from? The air pockets? It's the CO2. So when the bread rises as you are making bread, I've never made bread before, but I know that you have to let the bread rise, okay? As the yeast is breaking down the sugars in the dough, okay? That CO2 is being released, and it's gas that's causing the bread to rise. And when you bake the bread, it's the little air pockets that you see. And then let's talk about animals and some fungi. Some animals, such as us, and some fungi, when we don't have enough oxygen, we'll take that pyruvate and not break it down, but convert it into lactic acid. And just like with fermentation with bacteria and yeast, the pyruvate is going to be reduced. It's going to gain electrons from NADH. So the electrons from NADH are going to go to pyruvate. Pyruvate will be reduced, and you end up with a 3-carbon compound called lactic acid. Okay, and in some fungi and in some animals, they're going to use that NADH, NAD plus for glycolysis to make 2-ATP. This is how we make cheese. This is how we make, cheese. Some cheeses like blue cheese, right? You've got that fungi creating that blue cheese, okay? Or causing the lactic acid in cheese causes the proteins to like clump together, and that's how you get some of the cheese. That's something like that we did in class, right? We added vinegar to milk, and it caused the proteins in the milk to denature and clump up, and that's how we got cheese. But yeah, cheese is when we get that lactic acid that denatures the proteins in the milk, okay? And then sometimes when we are exercising or working out, we're not breathing in enough oxygen, so the pyruvate is not going to enter the mitochondria. It's actually going to stay in the cytosol where lactic acid will be produced, okay? So in greater detail, okay, that ethanol, okay, what does it look like? Okay, this is ethanol down here. So we've got the glucose and glycolysis, we end up with the 2-pyruvate. If there's no oxygen around, that pyruvate is going to become reduced. It's going to gain electrons, okay, and it's going to lose the carbon dioxide. And this is a molecule called acetaldehyde. How do you say that? Acetaldehyde. I don't know how to say it, okay? And that becomes ethanol. And it becomes ethanol when it becomes reduced, okay? It's going to gain electrons, and you get the 2-NAD, which can be used for glycolysis. So this regeneration of NAD is so important because it can be used for glycolysis, where the organism is going to make the 2-ATP. I can't believe I can't say the word. Let me spell it for you because it's down here and it's not shown in my shared screen. Okay, it's dehyde. Okay, acetaldehyde. Okay. Ethanol is toxic, right? And so that's why most bottles of like wine do not have more than 12% alcohol content, because if you get more than 12% or around 12%, it kills all the yeast. And so there's no more fermentation, no more alcohol being made, okay? And when ethanol is made, this is a chemical reaction that can't be reversed. You can't take ethanol and CO2 and bring it back together to make pyruvate, okay? That's what we call a dead-end process. Ethanol, CO2 made, that's it, okay? Whereas lactic acid is a reversible process, okay? It is reversible. So glucose is broken down in glycolysis to make 2-pyruvate. Pyruvate becomes reduced. It gains electrons from NADH, and you get lactic acid. Okay, you get lactic acid. Okay, so yeah, once O2 oxygen is available, that lactate can be converted back to pyruvate. Okay. Okay, so, we've been talking about glucose C6H12O6 as the starting point for cellular respiration in glycolysis, like glycolysis breaking down glucose. But actually, everything that you eat in your diet, like fats and proteins, they can also be broken down and they can enter this whole cellular respiration process anywhere within cellular respiration, okay? So for example, proteins, which are made up of amino acids, you can catabolize by catabolic reactions. You can break down the amino acids and it can actually turn into pyruvate. It can turn into acetyl-CoA. It can enter here in the Krebs cycle as any of those intermediates. And then you end up with NADH, right? NADH, you end up with FADH2s, you end up with ATP that's being made in the Krebs cycle, but then NAD and FAD are going to enter the electron transport chain to make ATP, okay? But then when you digest fats, they turn into glycerol and fatty acids. Glycerol can be converted into glyceraldehyde 3-phosphate, which can enter here in glycolysis, or the fatty acids can be broken down into acetyl-CoA, which will then enter the Krebs cycle. So really, whatever you eat, you don't have to start here with glucose. You can start with amino acids and glycerol and fatty acids. So one thing that's kind of cool, okay, about the whole production of ATP is that ATP can be a non-competitive or what we call an allosteric inhibitor. And previously we did talk about, when we talked about enzymes can be inhibited by inhibitors, right? They can be stopped by inhibitors. So it's interesting because ATP is an allosteric inhibitor of phosphofructokinase. And so it stops this whole glycolysis process early on so that more ATP is not going to be made. So this is just one of those examples of a biochemical pathway that is regulated by negative feedback, okay? So this is an example of negative feedback because negative feedback is when we stop a process from happening. Like this whole process of making ATP is stopped by ATP itself. It's almost, our cells are trying to, conserve energy by not making too much of something that is unnecessary, okay? Because if it's not necessary, you're just going to accumulate the product. So the product itself is going to act as an allosteric inhibitor. So it's really a balancing act of availability of the raw materials versus energy demands versus synthesis. So you don't want to make too much of something. And so ATP is an allosteric inhibitor of this enzyme, okay? And then also, that's an example, citrate, remember that was the first intermediate of the Krebs cycle. Citrate is also an allosteric inhibitor of phosphofructokinase, okay? Okay, so that is the end of our presentation, and I hope it wasn't too confusing. I just want you to understand that cellular respiration, remember the whole point is to make ATP, okay? And ATP is made in 3 parts of cellular respiration. You've got glycolysis. Let me just go back to this diagram, which I like. Glycolysis, Krebs cycle, and then the electron transport chain, okay? Understand that these NADHs and FADHs are so important, right? They're so important because they're going to be used to transport electrons to the electron transport chain where most of the ATP is going to be made in chemiosmosis. And you must know what chemiosmosis is, okay? All right, I hope that this video was helpful and that you learned a lot. And I hope that you have a really good day. Bye.